This is our project on single point mutation sickle cell. Here we see an ordinary DNA strand. Next we see the protein helicase unzipping the DNA strand. Next we have is the original sequence of the strand itself. Next, we have the single point mutation occurring. All the while, this is all occurring inside the nucleus. Once in the nucleus, the RNA is attempting to leave, and once it fully leaves the nucleus, it then becomes mRNA, messenger RNA.
As the mRNA leaves the nucleus and heads into the cytoplasm, it carries with it an adverse single point mutation. The single point mutation from A to T will eventually cause the entire genetic material to produce a different protein. While having just one copy of the mutated gene does not cause sickle cell anemia, having two copies does. Interestingly enough, one copy protects against malaria, an example of how mutations are beneficial. Here we see mRNA move through the cytoplasm. The single point mutation has altered the corresponding RNA molecule to GUG instead of GUA. The mRNA heads to the ribosome, where translation will now occur. As the tRNA constructs the corresponding amino acids, we ultimately end up with the mutant protein codon, VAL, instead of GLU. This is how sickle cell disease is constructed at the genetic level. At the protein level, we see here a picture of a normal protein versus the abhorrent clumped protein. As we draw even further to the cellular level, the single point mutation has affected the body's hemoglobin within the red blood cells. Hemoglobin, for those who do not know, is the iron-containing oxygen transport metalloprotein in the red blood cells of almost all vertebrates, as well as the tissues of some invertebrate vertebrates. Human hemoglobin in the blood carries oxygen from the lungs to the rest of the body. When red blood cells carrying mutant hemoglobin are deprived of oxygen, they become sickle-shaped instead of the usual round-shaped. These sickle-shaped red blood cells can get stuck in small blood vessels so that parts of the body don't get the oxygen that they need. This example illustrates how a single point mutation can have a large effect, in this case, both a positive and a negative one. But in many cases, evolutionary change is based on the accumulation of many mutations, each having a small effect. Whether the mutations are large or small, the chain of causation applies. Changes at the DNA level propagate up to the phenotype.
Recording. Sickle cell is very common amongst African Americans, and it connects to society. These are some of the symptoms: delayed growth, vision problems, anemia, pain in the joints and muscles, and repetitive infections. If these symptoms get worse, it can lead to a stroke, blindness, acute chest syndrome, or organ damage. Sickle cell is caused by the red blood cells clogging up. As you can see in the bottom blood vessel, they go to the shape of a sickle cell, and then when they try to go through a vessel, they will clog, but above is a normal free-flowing blood vessel. Sickle cell can be treated. Blood transfusions, high fluid intake with 8 to 10 glasses of water a day, receiving IV, or medications to help with the pain. Lastly, there is a gene editing that just came out, but it's only 20 to 40% effective. It turns a sickle cell back into a normal red blood cell. And then below in the DNA strand, what they do is they snip the A and T and replace it with a C and G strand. And that is our presentation on single point mutation of a sickle cell disease. Thank you, and we will appreciate the A+.